Gene expression is a process to convert the DNA into the protein. So in the next two chapters, for chapter 3 and chapter 4, we will discuss the detail about the gene expression. And then the first step of the gene expression is the transcription. So the transcription is a process to convert the DNA into the messenger RNA. And then the chapter 4, we will discuss this translation, which will convert the messenger RNA into the amino acid. And then first of all, we will watch the video about this detailed process.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So this is the process for the gene expression from the DNA to amino acid to the protein. And during this video, you can see the first step of gene expression is the transcription. And then for the transcription, So for the gene expression, we know the DNA is in the nucleus, but the protein is synthesized in the cytoplasm, and so they believe there is some type of molecule that can move the information from the DNA in the nucleus to the site of protein synthesis in the cytoplasm. And so there's a proposal that they believe there's a messenger in between. So the scientists they performed an experiment. They grew the bacteria E. coli for several generations on synthetic media that contain the heavy isotope, which has the nitrogen 15 and the carbon 13. As the bacteria use these heavy isotope carbon and nitrogen sources as raw material, so they can synthesize the carbohydrate, protein, and nucleic acid. All of their components became heavy labeled because they use the nitrogen 15 and carbon 13, and including the ribosome. They then change the genetic instruction by infecting the bacteria cell with the T2 bacteriophage virus. And the bacteriophage can destroy the bacteria DNA, substituting their own DNA as the genetic information that the bacteria would use to direct the synthesis of the viral protein. So if the ribosome carry the gene-specific information, then the new virus-specified ribosome should be made as well. If, on the other hand, the ribosomes are just passive sites of synthesis, the old bacterial ribosome could be used by the virus commanded cell to make the virus-directed protein. The distinction here concerns the source of the information in protein synthesis, which occur on the ribosome. If the information could be shown not to reside in the ribosome, then it had to have been transported there by some other element, which is the messenger. So the radio-labeled phage RNA in experiment was found to be associated with all the ribosome. As you can see that here, the all ribosome that carry associated with the radio-labeled phage RNA. Therefore, the ribosome RNA was made before the infection, and ribosome RNA does not carry information from DNA. A different class of unstable RNA associated transiently with ribosome. So that confirms there's a messenger RNA in between the DNA and the protein. So that's the early experiment to determine and confirm the presence of messenger RNA. Transcription is the first stage in gene expression, and then the transcription converts the DNA into the messenger RNA. And as we know, DNA is double-stranded, so there's one coding strand and then there's a template strand. So the messenger RNA basically is made through the template strand. Use the template strand to make the complementary strand of messenger RNA, and the messenger RNA sequence will be exactly the same as the coding strand. Here's another video to show the transcription.
DNA's most extraordinary secret. How a simple code is turned into flesh and blood.
How a simple code
It begins with a bundle of factors assembling at the start of a gene.
It begins with a bundle of factors.
A gene is simply a length of DNA instructions stretching away to the left.
A gene is simply a length of DNA instructions stretching away to the left.
The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein.
Reading off the information that will be needed to make the protein.
Everything is ready to roll. 3, 2, 1, go.
Everything is ready to roll. 3, 2, 1,
The blue molecule
go.
racing along the DNA is reading the gene.
The blue molecule racing along the DNA
It's
is reading
unzipping
the
the double
gene.
helix and copying one of the two strands.
It's unzipping the double helix and copying one of the two strands.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA.
And it's made of a close chemical cousin of DNA called
The
RNA.
building blocks to make the RNA enter through an intake hole.
The building
They are
blocks to make
matched
the
to the
RNA enter
DNA,
through
letter
an
by
intake
letter,
hole.
to copy the A's,
They are matched
C's,
to the
T's,
DNA,
and
letter by
G's
letter,
of the
to
gene.
copy the A's, C's, T's, and G's
The only
of the
difference
gene.
is that in the RNA copy, the letter T is replaced with a closely related building block known as U.
The letter T is replaced with a closely related building block known as U.
You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
It's happening right now.
So this is the process of transcription to convert the DNA into the messenger RNA using the template strand to make the complementary messenger RNA. A transcription unit is a region from the promoter to the terminator. So as you can see from this slide, we have the promoter and we have transcription starting side and all the way to the terminator. And this piece we call transcription unit. And in eukaryotic cell, a gene may contain internal regions that are not represented in the protein. Therefore, we have exon, intron in the DNA. And when we transcribe into the messenger RNA, the pre-messenger RNA, and then this intron can be spliced out, and so that we connect those exons together to make a mature messenger RNA. And then this process, we call it the RNA processing, which we will discuss further later. And then this messenger RNA will be transported into the cytoplasm, and then in the ribosome, and then translate into the protein. So that's the process in the eukaryotic cell. So as we know, the transcription is an RNA synthesis process, and for the RNA synthesis process, we need the enzyme to catalyze the reaction. And so for the transcription, we need the RNA polymerase to catalyze the phosphodiester bond formation, and it is very similar to the DNA polymerase action to synthesize the DNA. So basically, again, the hydroxy group will attack this alpha phosphate and then remove the pyrophosphate and then form this phosphodiester bond. So that's how they form the phosphodiester bond in the RNA synthesis. For the RNA synthesis, it requires several components to perform the reaction. And first of all, we need a DNA templates, so they can act as the template and then to form this complementary RNA sequence. Again, in here, we use the RNA synthesis, use the uracil to replace the thymine. And so we require the ATP, CTP, GTP, and the UTP for RNA synthesis. And RNA chain grow in the 5' to 3' direction, just like the DNA synthesis. And the only difference is RNA polymerase does not require the RNA primer. Remember that when we discuss the DNA synthesis in the DNA replication, it always requires the RNA primer to provide the hydroxy group to initiate the DNA synthesis. But for RNA polymerase, does not require the RNA primer. So for the promoter, here we can see how they number the nucleotide. So for the first nucleotide, for the transcription start side, we number as the +1, and then to the right would be the downstream, so +1, +2, and then before the transcription start side, that will be the upstream sequence, so -1, -2. So that's how they number the RNA sequence. And so top strand, that's the coding strand, and bottom strand is the template strand. And here, that's how they synthesize the new RNA strand. So for the promoter, so the promoter basically is the region where the RNA polymerase will recognize and bind so that they can initiate the transcription. So for the prokaryotic cell, we have the two important components. One is the -10, the consensus sequence, Tata sequence, and also -35. So these are important promoter consensus sequence for RNA polymerase to recognize -10, -35. And for eukaryotic promoter, it will be more complicated depending on these different polymerase, because in eukaryotic cell we have RNA polymerase 1, polymerase 2, polymerase 3, but mostly that for the messenger RNA is a polymerase 2. And for polymerase 2, we have -25 Tata box, which is very important consensus sequence for the RNA polymerase 2 to recognize. And then for the bacteria, for the prokaryotic cell, and some of them have a very strong promoter. For the strong promoter, they have this extra the UP up elements, right? So we have -10, -35, that's a core promoter. Then further upstream, we have this up element between the -40 and -60. And later on, we will discuss the importance of the up elements. RNA polymerase is a complex protein. It has several subunits. So as here you can see from this SDS page gel to show you the different subunit in the RNA polymerase. It has the 2 alpha subunit, and the 1 beta prime, 1 omega, and 1 sigma. And in the next few slides, we will discuss each of these subunits' function for the during the transcription. The RNA polymerase, and we have to know there are coenzyme and the holoenzyme. It depends on the component of the enzyme. So if we have the 2 alpha subunit, 1 beta and beta prime together, we call it a coenzyme. And if we add sigma, then that would be the holoenzyme, which would be a complete RNA polymerase. So basically, to initiate the transcription, it will require this holoenzyme to initiate the transcription. And to continue the RNA synthesis, it just requires this coenzyme. And then the role factor basically relevant to the termination. And here just to show you that different subunit, we have the 2 alpha subunit basically is encoded by the RPO-A gene, and the beta subunit is RPO-B, beta prime is RPO-C, and the sigma is RPO-D genes. So the holoenzyme can be separated into the 2 components as we described earlier. So we have coenzyme and the sigma factor. And then only the holoenzyme can initiate the transcription, which is very important. Without the sigma factor, the RNA polymerase will not be able to initiate the transcription. And the function of a sigma factor basically is to bind in a stable manner to the DNA at the promoter, because the sigma factor can direct the coenzyme to the core promoter so that they will be able to initiate the transcription. And the coenzyme only has the ability to synthesize RNA on the DNA template, but they cannot initiate the transcription. And here is just to show you that the diagram, the sigma factor control the specificity, because the coenzyme, they can scan through the DNA strand, but only in the present sigma factor they can direct the coenzyme to the promoter, and so that they can initiate the transcription. And this is just an example to show you the binding of the DNA to the holoenzyme is very stable, and if with the coenzyme, with the time, and then they would reduce the binding to the coenzyme. And also the sigma factor has been confirmed that can be reused. So from this slide you can see the sigma bind to the coenzyme, direct the holoenzyme to the promoter region, initiate the transcription. Then the sigma factor will dissociate from the coenzyme, and the sigma can also reuse through the binding to another coenzyme to initiate another transcription. So the sigma factor can be recycled. Slide demonstrates that the sigma factor bind to the coenzyme to form the RNA polymerase holoenzyme, so that they can direct the holoenzyme to the promoter region, because this is the proposal proposed by the Hinkle and the Chamberlain. So they believe the RNA polymerase they bind to the DNA loosely at first, and then they will scan along the DNA until they find a promoter, so that the holoenzyme will bind to the promoter very strongly, and then in the beginning is a closed promoter complex, and then the holoenzyme can mail a short DNA region at the promoter to form an open promoter complex with the polymerase, and then they can initiate the transcription. Transcription occurs in 3 phases. The first phase is initiation. The second phase is elongation, and then the termination. So for the initiation, RNA polymerase binds to the promoter tightly and melting and opening the DNA sequence that will be transcribed. And then the first phosphodiester bond of the new RNA formed using the template strand. And then that's the initiation. Once the first phosphodiester bond of new RNA formed, then the bubble, the bubble, which is the open area, will move with the polymerase, allowing it to read and transcribe the DNA continuously, and now will enter the elongation stage. At this stage, the sigma factor will dissociate from the holoenzyme, so the coenzyme will continue the elongation stage until the termination. So for the initiation, so the RNA synthesis begin at the promoter, and the first base of synthesized RNA is a +1, and then as you can see here, we have the transcription bubble, because a typical example of transcription bubble consists of 14 unpaired bases, and the first 9 have been used to transcribe new single-stranded RNA. And as the RNA polymerase synthesize the RNA, then the RNA polymerase will move and continue to open the double helix DNA and then transcribe the DNA. Initiation begins when RNA polymerase bind to the promoter and form the closed complex. As you can see here, from the closed complex on the slide, the sigma subunit directs the polymerase to the promoter by binding to specific sequence upstream of the start site, which is the promoter sequence, and which is that the -10, -35, those consensus sequence. And then after the binding of the sigma subunit to direct the polymerase to the promoter region, both beta subunit and the sigma subunit, they initiate separation of the 2 downstream DNA strands, melting about 10 to 17 base pairs surrounding the transcription start site to form the open complex. The open complex, that will be the transcription bubble. And next, the polymerase start building the RNA chain. The first base in the RNA is a purine ribonucleoside triphosphate, and the A tend to occur more often than the G. After the first nucleotide is in place, the polymerase joins a second nucleotide to the first, forming the initial initial phosphodiester bond in the RNA chain. Both beta and beta prime subunit are involved in the phosphodiester bond formation, as well as the DNA binding. Several nucleotides may be joined before the polymerase leave the promoter and elongation begin. So when the elongation begin, the sigma subunit will dissociate from the RNA polymerase. And within the transcription bubble are the 9 most recently added ribonucleotide of the RNA transcript. So the transcription bubble, basically transcription bubble, just the transiently, the DNA double strand transiently separated into its single-stranded, so that the polymerase, RNA polymerase, can use the template strand to synthesize RNA. So that's the transcription bubble. So how do we determine the presence of transcription bubble and the size of transcription bubble? Basically, we use this chemical reagent, dimethyl sulfate, DMS, because DMS would be able to transfer the methyl group to the adenine, and therefore the AT will be no longer able to form the base pair, and so that will be a bodge, and then that can be cleaved by the enzyme. So here you can see if we have this double-stranded DNA, and then we have AT base pair, and once we melt and then we add the DMS, the DMS will transfer the methyl group to the A, and therefore the A and T will not be able to form the base pair, and so that will give you this bodge, and that can be cleaved by the S1 nucleus. And once they cleave, the S1 can find those base paired AT and cleave it, and from this cleavage we produce several different fragments, different size fragments, because the cleavage can cleave as, for example, the first AT pair, second AT pair, or third AT pair, and produce different size fragments. Then after the geolithophoresis, we'll be able to determine the location of those AT base pair that has been opened, and then added the methyl group. So in this actual geolithophoresis that you can see in this lane here, so we have these experiment that we add the RNA polymerase, and then we add the DMS and also S1 nucleus. So you can see this band here. This band basically are the bands that showing the area where the transcription bubble is. So that will be between the -9 to +3. That indicate this is the area, the region where the RNA polymerase bind and initiate the transcription bubble. And the role of the alpha subunit, as I mentioned earlier, the up element, that will be the very strong promoter. So basically the up element can be recognized by the alpha subunit, okay? Because sigma factor recognize the core promoter, and then the alpha subunit can help to recognize the up UP element. And this is an example to show you that in vivo transcription on the plasmid containing the promoter. And then we have up element, and then we can see in the presence of the alpha subunit, they can help to recognize this region, the up element, and then help to increase the transcription efficiency. And so once the RNA polymerase bind the promoter region and then initiate the transcription, so that the promoter region will be open, and then once they move along the template, and the promoter region will close. Once they enter the elongation stage, the sigma subunit will leave the RNA polymerase. So the next stage is elongation. So that elongation, basically the nucleotide are added sequentially, one after another, in the process of elongation. And this elongation stage only need the coenzyme, which means at this stage the sigma subunit dissociate from the RNA polymerase, so it becomes the coenzyme only has 2 alpha subunit, 1 beta and 1 beta prime subunit, and so they can continue to transcribe the template. And this is just to explain during the elongation, basically the phosphodiester bond formation reaction is catalyzed by the beta and beta prime subunit. And for the topology, as we discussed in chapter 1, when you open the double helix DNA, that will introduce the tension to the double-stranded DNA. And in this case, for the transcription bubble, when you open up double helix, that will introduce the positive supercoil ahead of the RNA polymerase, and then negative supercoil behind the RNA polymerase. And the positive supercoil will require topoisomerase to unwind the positive supercoil. And during the transcription, RNA polymerase frequently pause, even backtrack during the elongation. This is because the pausing allows the ribosome to keep pace with the RNA polymerase, and it is the first step in termination. And backtracking can help to proofread the newly synthesized RNA if it's the wrong nucleotide being incorporated into the RNA strand, then they can be removed, and then they can backtrack and then resynthesize the correct RNA. After the elongation, finally will be the termination. So when the polymerase reaches the terminator at the end of a gene, it falls off the template and releases the RNA. And there are 2 main types of terminator. One is the intrinsic terminator. Intrinsic terminator basically is the sequence itself. So as you can see on the slide, for the intrinsic terminator, you will see the inverted repeat sequence to form the inverted repeat sequence will be GC reach sequence. They can form the helping stem, and then there will be a loop, and then in the end there will be the poly U sequence, and then there will be terminus. And so basically this is a low independent termination, which will be the intrinsic terminator. So when the RNA polymerase reach to this region, then the RNA polymerase will fall off from the template, and the poly U is about 7 to 9 bases for poly U. And then the second mechanism is a rho-dependent mechanism. So for the rho-dependent mechanism, there is no string of the poly U in the end of the sequence. It's just the inverted repeat to helping, and binding to the growing transcript. The rho follow the RNA polymerase. It catches the polymerase as a pause at the hairpin, and release transcript from the DNA polymerase complex by unwinding the RNA-DNA hybrid. So that's the rho-dependent termination. Our genome has more than 3,000 genes. Some of these are active all the time because their products are in constant demand. But some of them are not transcribed most of the time because their products are rarely needed. The reason that the cell does not leave all its gene on all the time is because gene expression is an expensive process. It takes a lot of energy to produce RNA and protein. Therefore, control of gene expression is essential to life. And for the E. coli, there are several mechanisms several strategies to regulate the transcription. So they could be here are the 7 major strategies: alternative sigma factors, RNA polymerase switching, anti-termination, enhancer, operon, transcription attenuation, and the ribose switch. And so for the alternative sigma factor, RNA polymerase switching, and the anti-termination, these 3 strategies involve the change of transcription machinery. And the operon, transcription attenuation, and the ribose switch involve the regulation of group of functionally related genes. Enhancer are the DNA element that bind protein factors. For the alternative sigma factor, so basically when the phage infects bacteria, it usually subverts the host transcription machinery to its own use. In the process, it establishes a time-dependent or temporal program of transcription by producing different sigma subunits that direct the RNA polymerase to different genes. So that's why it's called alternative sigma factor. So one of the examples would be the SPO1. SPO1 is a virus that infects the bacteria, Bacillus subtilis. The virus has a set of genes called early genes, which are transcribed by the host RNA polymerase using its regular sigma factor. One of the viral early gene codes for protein GP28, which is a sigma subunit. This sigma subunit directs the RNA polymerase to transcribe more of viral genes in the middle phase. Products of the 2 middle genes are GP33 and GP34, which together make up another sigma subunit that direct the transcription of the late genes. In the end, viral sigma subunit control the transcription machinery for the virus instead of the bacteria. So this is an example of alternative sigma subunit. And when bacteria cells experience an increase in temperature or a variety of other environmental insults, they mount a defense response called a heat shock response. To minimize damage, a set of heat shock genes are expressed to protect the cell. In this situation, the heat shock response in E. coli is governed by an alternative sigma subunit, sigma 32, which displaces sigma 70 and directs the RNA polymerase to the heat shock gene promoter. In addition to the alternative sigma subunit, the sigma 32 mechanism, E. coli has evolved ways of controlling transcription using anti-sigma subunit when cells are stressed by such insult as loss of a nutrient, high osmolarity, or high temperature. They stop growing and enter the stationary phase. At this point, there's a protein called RSD protein is produced. It can bind to the sigma 70 subunit and block its binding to the core polymerase. Therefore, this anti-sigma subunit can supplement the sigma replacement mechanism by inhibiting the activity of sigma 70 subunit activity. So this is during the environmental stress. For the RNA polymerase switching, again, it's another bacteriophage. So the bacteriophage T7 is a simple E. coli phage, and it has 3 phases of transcription: an early phase called class 1, and 2 late phase called class 2 and 3. Instead of making a new sigma subunit like the SPO1 for the alternative sigma subunit, a specific class 1 gene encodes a new RNA polymerase with absolute specificity for the class 2 and 3 genes of phage T7. And therefore, the switching mechanism in this phage is very simple. So that's RNA polymerase switching. Then another strategy is anti-termination. Anti-termination is a regulatory event because termination is prevented when anti-termination protein act on the RNA polymerase. This causes to read through a specific terminator or terminators, because this is also a mechanism in the bacteria to prevent the bacteriophage's infection. And then the bacteriophage has this anti-termination mechanism to prevent the bacteria's inhibition. So one of the examples is the phage, lambda phage infection. In the lective mode, almost all of the phage genes are transcribed and translated, and the phage DNA is replicated, leading to the new progeny. And then in the lysogeny mode, the lambda DNA is incorporated into the host genome. And only the CI, which encodes the lambda repressor protein, is expressed and then prevent the transcription of all the rest of the phage gene. One of the two immediate early gene is the CRO, which coded for the repressor of the CI gene. That's a CRO, coded for a repressor of the CI gene that allow the lytic cycle to continue. And the other one is called N, coded for the anti-terminator, N that override the terminator after the N and the CRO gene. Transcription then continue into the delay early gene. One of the early delay gene coded for another anti-terminator that permit the transcription of the late gene. This anti-terminator acts to alter the polymerase, so it reads through the terminator to continue to the transcription of gene in the next phase. So that's the anti-termination. The operon. Operon basically is a group of a contiguous coordinated controlled gene and in E. coli. This is because in prokaryotic cell, genes that encode enzyme of certain metabolic pathway are often controlled as a group, with the gene encoding the protein of the pathway being close together and under the control of a common promoter. So that's operon. And the expression of this gene can be triggered by the presence of a suitable substance called inducer. And this phenomenon is called induction. Because all genes in an operon are transcribed together to produce one message, it is also called polycistronic message. And here is just the several different scenarios for the regulation of gene expression. So for example, it can be a negative control and positive control. For the negative control, for the induction, basically, you have the repressor. Repressor can bind to the promoter to inhibit the transcription, but in the presence of co-inducer, and then the expression can be induced. And basically, this is a negative control because under normal circumstances, the repressor binds to the promoter to inhibit the transcription. And then in the presence of co-inducer, that expression can be induced. And now be the example be lactose operon. And then for the tryptophan operon, it's also negative control by the repression. So basically the inactive repressor can be produced, but it will not bind to the promoter. Only in the presence of co-repressor, then the repressor become active and bind to the promoter and inhibit the transcription. So that would be the tryptophan operon. And then this is repression, negative control. For the positive control, on the right side, the catabolized repression. So normally the inducer is produced by its inactive. Only in the presence of the co-inducer, then they will bind to a promoter and then induce the transcription. And then here will be the positive control for the repression. So basically the active inducer binds to the promoter for the expression, but in the presence of co-repressor that can bind to the active inducer, and then the inducer become inactive, so there'll be repression. And here is just to show you different type of gene expression: negative regulation versus the positive regulation. So in the bottom, here's a table. So for the positive regulation, when there's a binding of the regulator, then the expression is on. When there's no binding, it's off. And for the negative control, if there's a binding, then the transcription is off. And then if there's no binding, the transcription is on. So that's the negative control versus the positive control. And this is the typical example of negative control of the leg operon. So in the panel A, when there's no lactose, the leg I gene produce the repressor, and the repressor bind to the operator, and the operator basically overlap with the promoter region. Therefore, the RNA polymerase will not be able to bind to the promoter, so no transcription. And then in the bottom, the panel B, in the presence of lactose, there'll be derepression. In the presence of lactose, the inducer will bind to the repressor, and the repressor change the conformation. Therefore, the repressor cannot bind to the operator, so that the RNA polymerase can bind to the promoter and then initiate the transcription. So this is an example of a negative control. And this is the positive control by the CAP. The CAP site, which is the enhancer. So basically, both the glucose and lactose are present. The cell does not make the leg protein. The repression of the synthesis of the leg protein by glucose is called catabolite repression, that involves two regions in the promoter. One is the binding site for the RNA polymerase, and the other is the binding site for the another regulatory protein, the catabolite activated protein, CAP. When the glucose is not present, CAP form a complex with the 3', 5' cyclic AMP, CAMP, and bind to the CAP site on the promoter. This binding helps the RNA polymerase bind to the polymerase, and operon is fully active. In the presence of glucose, which is the repression, the level of CAMP is low. Only very few CAP cyclic AMP complex can be formed. It is not enough to activate leg operon. So the presence of lactose is necessary for the transcription of operon, but not sufficient. The leg operon is active only when the glucose is absent and the lactose is present. The leg operon is active only when the glucose is absent and the lactose is present. Therefore, the CAP site is an example of an enhancer element, and the CAP cyclic AMP complex is a transcription factor. An enhancer is an element that stimulates transcription and are cis-actin DNA elements that are not strictly part of a promoter, because it could be very far away from promoter, can be up to several kilobase distant, and either upstream of or downstream from the promoter they control. The regulation is done by looping the DNA around so that the activated protein bound at the enhancer can make contact with the transcription machinery through a protein non-mediated complex, and so that they can activate the transcription. So that's the enhancer. And the transcription attenuation. So basically the example of the trip operon. The trip operon responds to a repressor protein that binds to the two-molecule tryptophan. When the tryptophan is plentiful, this repressor tryptophan complex binds to the trip operator, and this binding prevents the binding of RNA polymerase, so the operon is not transcribed. On the other hand, when the tryptophan levels are reduced, the repressor will not bind the operator, so the operon is transcribed. This is an example of a system that is repressible and under negative control. In addition to the standard negative regulation, the trip operon is regulated by another mechanism of control called transcription attenuation. This mechanism operates by causing a premature termination of a transcription of the operon when tryptophan is abundant. There are two loci, the trip leader and the trip attenuator, in between the operator and the gene TRPE. Secondary structures formed in the messenger RNA of the leader sequence are responsible for this premature termination. The formation of such secondary structure comes from the transcription stop signal, an inverted repeat, and a string of 8 AT pairs in the attenuator. When it is plentiful, transcription is terminated prematurely after the leader sequence has been transcribed. Therefore, attenuation imposes an extra level of control on an operon over and above the repressor operator system. So that's the transcription attenuation. For the ribose switch, a ribose switch is a region usually in the 5' untranslated region of a messenger RNA that contains two modules, an atomer and an expression platform. An atomer is a region that binds to a ligand. On the other hand, this expression platform can be a terminator, a ribosome binding site, or another RNA element that affects gene expression upon the change in conformation. For example, the RIP-D operon in the Bacillus subtilis controls the synthesis and transport of the vitamin riboflavin and one of its products, flavin mononucleotide, FMM. And the RIP operon contains a conserved element in their 5' untranslated region known as the RFN element, which is an atomer. There are two alternative conformations of the RFN element, anti-termination and the termination conformation. FMM binds directly to the RFM element and causes it to form the termination conformation, blocking the expression. In the absence of FMM, the element forms an anti-termination conformation, which can complete the transcription. So that's a ribose switch. Now we will discuss the transcription in eukaryote. So the transcription process is more complicated in eukaryote. 3 RNA polymerases with different activities are known to exist, and each one transcribes a different set of genes and recognizes a different set of promoters. RNA polymerase 1 synthesizes precursor of most ribosomal RNA. So you can see here large ribosomal RNA precursor, 28S, 18S, and 5.8S ribosomal RNA. RNA polymerase 2 synthesizes the messenger RNA precursor. RNA polymerase 3 found in the nucleoplasm and synthesize the transfer RNA, precursor of 5S ribosomal RNA, and some small RNA molecule involved in messenger RNA processing and protein transport. So all 3 eukaryotic RNA polymerases are large complex and contain 10 or more subunits. They are all related to the prokaryotic polymerase and to one another. And so, for example, 3 polypeptide, RPP1, RPP2, RPP3 are absolutely required for enzyme activity in yeast, and they are homologous to a beta prime, beta, and alpha subunit in E. coli. And both RPP1 and beta prime subunit bind the DNA. RPP2 and beta subunit are at or near the nucleotide joining active site for the synthesis. And then similarity between the RPP3 and alpha subunit. There's a 20 amino acid subunit of great similarity two subunits about the same size, and 2 monomer per holoenzyme. And 3, because they are 3 RNA polymerase, so they will require different promoters, right? Because they have a different structure and then transcribe different class of gene, so that they have different they have different promoter. So for class 2 promoter, so the core promoter can attract the general transcription factor and RNA polymerase 2, and then therefore they determine the start site and direction of transcription. And for the class 2 promoter, they are core promoter and proximal promoter. And proximal promoter help to attract the general transcription factor and RNA polymerase, and include promoter element upstream of the transcription start site. And in the core promoter, so the core promoter, we have the Tata box, initiator, downstream element, upstream element. And here is just in this slide, you can see the location of these elements in the core promoter. We have this Tata box and around the -25, upstream element it's about -40 to -91, including the GC box, initiator, that's a +1, and also we have downstream element. So the initiator box is a sequence found at the site where the transcription starts, and then -25, that's Tata box. That's the place where the Tata binding protein will bind. And GC box has a consensus sequence GGG, CGG. They bind the transcription factor SP1. And the proximal promoter are usually found the upstream of the class 2 core promoter. They differ from the core promoter in binding to relatively gene-specific transcription factor, which are the transcription activators. So these are the specific feature for the class 2 promoter. Then for the class 1 promoter, basically that is bounded by the RNA polymerase 1. The class 1 promoter are now well conserved in sequence across species, and they have the core element surrounding the transcription start site. The core element, transcription start site, of course, they're bound by the TBP, Tata binding protein, and SL1, and also the upstream control element, UCE element, which is 100 base pair farther upstream. And class 3 promoter. So the classical RNA polymerase 3 promoter are type 1 and type 2, which have promoter lie wholly within the genes. Both type 1 and type 2 genes include a variety of RNA genes, such as the transfer RNA, the 5S RNA subunit of ribosome, and the adenovirus VA RNA gene. And the type 3 RNA polymerase 3 promoter is non-classical, and it resembles those of the class 2 gene, including U6 snRNA gene, and 7SL RNA gene, and 7SK RNA gene. So these are class 3 promoters. And the order of event in eukaryotic transcription initiation. So eukaryotic RNA polymerase are not capable of binding by themselves to their respective promoter. They rely on the proteins called a transcription factor to bind to their promoter. A transcription factor is any protein that regulates transcription by itself is not a subunit of the RNA polymerase, and that's called a transcription factor. And there are two types of transcription factor: the general transcription factor and the gene-specific transcription factor, which is the activator. So the general transcription factor combine with the RNA polymerase to form a pre-initiation complex that's competent to initiate transcription. Although the general transcription factor can supply only a basal level of transcription in the absence of activators, the pre-initiation complex is the most important step to start the transcription. Furthermore, this is known about the elongation termination in eukaryote than in prokaryote. So we just discussed the sequence of events of the initiation in RNA polymerase II transcription as an example to understand the eukaryote transcription. So the first step of the transcription is the binding of the TF2D to the promoter. So the TF2D, with the help from the TF2A, bind to the Tata box, forming the DA complex. And then the TF2B bind next to generate the DAB complex. TF2B is critical for the assembly of the initiation complex and for the location of a correct transcription start site. And subsequently, the TF2F binds tightly to RNA polymerase II, and this complex binds stably to the promoter. Finally, the TF2E and TF2H bind to RNA polymerase II to form the complete pre-initiation complex. To start the transcription, the RNA polymerase II is phosphorylated in the C-terminal domain, and then the DNA is separated, and then they start the transcription. The primary protein of TF2D is the Tata binding protein, the TBP, and it is associated with many TBP associated factor, TAF2S. And the transcription factor, the transcription activators. So basically the activator, they can bind to the DNA, and so they have a specific domain, a motif that can help the activator to bind to the DNA. So there are three different classes of DNA binding motif: the zinc containing a module, which is zinc finger, and homeo domain, and the B-zip and the BHLH motif. And the zinc fingers are one of the zinc containing module. It is composed of an antiparallel beta sheet followed by an alpha helix. Two cysteine from the beta sheet and two histidine from the alpha helix are coordinated to a zinc ion. This coordination of amino acid to the metal helps form the finger shape structure. On the other hand, some zinc containing DNA binding domain do not have zinc finger. For example, the GAL4 protein does not have zinc finger, but its DNA binding motif has 6 cysteine that coordinate to zinc ion in the bimetal thiolate cluster. Homeo domain contains about 60 amino acids and resemble in structure and function the helix-turn-helix DNA binding domain of prokaryotic protein such as lambda-Faj repressor. Each homeo domain contains 3 alpha helix. The second and the third of these form the helix-turn-helix motif, while the third serves as the recognition helix. The B-zip protein dimerized through a leucine zipper, so they can interact with the DNA. Epigenetic regulation of eukaryotic transcription is another mechanism that can regulate the gene expression. This is because the eukaryotic DNA is packaged into the chromatin, and the basic structural repeat unit is the nucleosome. And in the nucleosome, we have 147 base pair of DNA wrapped in about 1.75 super helical turn around a central octamer composed of 2 each of the 4 core histones. H2A, H2B, H3, and H4. And the histone octamer does not bind to the DNA randomly. So basically, they exhibit preference for some sequence over others, and we call that nucleosome positioning. And because of the location of this nucleosome, they can control and regulate the gene expression, because RNA polymerase has to go through this DNA region for transcription. So that's why this histone and nucleosome may influence the gene transcription, and this regulation we call epigenetic regulation. So the first epigenetic regulation mechanism is the histone modification. So covalent modification of histone is important because the DNA is negative charge, and then in the N-terminal of the histone, those are co-histones. They have several positive charged amino acids. These positive charged amino acids can interact with the DNA tightly. So if we modify those positive charged amino acids, then the interaction between the histone tail and the DNA is no longer exist, and then the RNA polymerase will be easy to access to those DNA. So that's the first mechanism of the epigenetic regulation, and also the histone modification can also provide the histone code for the regulation. And the second mechanism would be the chromatin remodeling complex. The chromatin remodeling complex basically can change the nucleosome position throughout the transcription process. Therefore, the RNA polymerase will be able to access the DNA easily. So that's the second mechanism of the epigenetic regulation of gene expression. And the histone code basically that would be the memory of the DNA, the cell, to have those histone modification and the chromatin remodeling activity, so that they can direct the transcription. So that's histone code. And the transcription elongation through the nucleosomal barrier. This is because the chromatin is remodeled upon the transcription initiation. The DNA remains packaged in nucleosome in the coding region of the transcribed gene. As the RNA polymerase II moves through the gene coding region, the enzyme encounters the nucleosome approximately every 200 base pair. RNA polymerase II overcomes this nucleosomal barrier through the existence of two distinct mechanisms for the progression of RNA polymerase through the chromatin. Number one is the nucleosome mobilization, which is the octamer transfer. So they can, as the RNA polymerase II goes through the coding region, so the nucleosome can be mobilized. So that means they can transfer octamer to another region, so that the RNA polymerase can go through it easily. Or the H2A, H2B dimer depletion. So the depletion of H2A, H2B can make the nucleosome structure is loose, and then the RNA polymerase can go through it easily. And this is the video to show the epigenetic regulation and the associated product offering.
Epigenetic regulation encompasses a number of different modifications to chromatin. These include methylation of the DNA on cytosine bases, a modification that can further be oxidized, as well as modification of the histone tails that emanate from the core of the nucleosome. The tails of core histones labeled here can be altered with distinct chemical modifications, including methylation of histone H3, acetylation of histone H4, and phosphorylation of histone H2B. Euchromatin is often characterized by a more open and accessible state of the DNA, one in which transcription factors have access to their cognate binding sites and can therefore recruit enzymes like histone acetyltransferases that acetylate histone tails and activate genes by recruiting components of the basal transcriptional machinery, including RNA polymerase. Heterochromatin, in contrast, is thought to be characterized by a more repressive, tight bundling of nucleosomes, which impedes transcription factors from gaining access to regulatory sites on the DNA. Methylation of cytosine bases in regions called CPG islands is a hallmark of transcriptionally repressed heterochromatin. These methylated cytosines in turn recruit proteins like MECP2, methyl CPG binding protein 2, and HP1, heterochromatin protein 1. These proteins are thought to maintain a repressive state of chromatin by inducing histone deacetylation by HDACs, as well as histone tail methylation by histone methyltransferase enzymes.
So this is the epigenetic regulation of transcription. And the next section now we'll discuss the post-transcriptional modification messenger RNA. The genes of prokaryote are continuous. Every base pair in the continuous prokaryote gene is reflected in the base sequence of messenger RNA. The genes of eukaryotes are not necessarily continuous. Instead, eukaryotic genes frequently contain intervening sequences that do not appear in the final messenger RNA for that gene produced. The DNA sequences that are expressed are called exon, and the intervening sequences are called intron. As such, the expression of eukaryotic gene involved not just its transcription, but also the processing of the primary transcript to its final form. And that's the first modification, which will be the RNA splicing. And then also there are two other mechanisms after the post-transcriptional modification for the post-transcription modification. One is the 5' cap, and another one is polyadenylation of the 3' tail. So that would be the three mechanisms can be processed after the transcription is completed. So for the 5' cap, the 5' cap basically is the cap at the 5' end of eukaryotic messenger RNA. On the left of the panel, you can see a simplified process. So at the 5' you have triphosphate, and the first step is to remove one phosphate, and so that you keep the diphosphate. So that requires RNA triphosphatase to remove the one monophosphate. That's the first step. And then the second step is the addition of the GMP, and this requires the RNA guanine transferase to transfer a GMP to to connect to the diphosphate area. And so when the GMP, the guanine monophosphate, attached to the diphosphate, then you will see the triphosphate again. And so that's the addition of a GMP required RNA guanine transferase. And the last step is methylation of the added guanine. So that requires the guanine N7 methyltransferase to transfer a methyl group to the N7 in the guanine, in the 7-methyl guanosine. So that will be the 5' cap. The 5' cap. The 5' cap is essential for the efficient elongation and termination of transcription, as well as the subsequent processing of messenger RNA. It also functions as the binding site. The 5' cap occurs soon after the growing RNA emerges from the RNA polymerase, and then it is accomplished in these three steps: the removal of 5' phosphate, addition of GMP, and methylation of added guanine. Polyadenylation of the 3' end of the eukaryotic messenger RNA begins with an initial cleavage of the messenger RNA. The cleavage is usually occur after a CA dinucleotide pair that lies somewhere between a conserved AAU-AAA hexamer sequence and a U or GU rich region further downstream. After cleavage, a tail of approximately 200 adenosine is added by poly-A polymerase. This process is more complex than the 5' capping because it involves recognition and processing of different polyadenylation sites in different messenger RNA. And the splicing, the mature transcript for many genes is encoded in a discontinuous manner in a series of discrete exons. RNA include messenger RNA, transfer RNA, and ribosomal RNA, all contain introns that must be removed from precursor RNA to produce functional molecules. tRNA precursor undergo splicing that is catalyzed by protein factor, and ribosomal RNA precursor catalyze their own removal and are termed self-splicing. On the other hand, messenger RNA splicing is carried out by the spliceosome. Spliceosome is composed of several individual small nuclear ribonucleoprotein snRNP and the many more additional proteins that come and go during the splicing reaction. snRNP remove the intervening sequence from pre-messenger RNA, and they contain a large number of proteins and the 5 small nuclear RNA, snRNA, including U1, U2, U4, U5, and U6 snRNA. They are post-transcriptionally modified protein. The snRNA of the snRNP recognize 3 sites on the pre-messenger RNA. These are the 5' splice site, 5' AG, GU, AAGU sequence, and then the 3' splice site. So that would be the YY, 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 NCAG. Y is any pyrimidine, N is any nucleotide. And then the third one is the branch site, which will be the UACU AAC. The vast majority of the intron begin with GU and end in AG. Recognition is due to base pairing between the snRNA and the primary transcript. The protein part of the snRNP supervises the cutting and joining reaction. The snRNP assemble onto the pre-messenger RNA, forming the spliceosome, and then the SNRP assemble onto the pre-messenger RNA, forming the spliceosome. Recognize the 5' splice site, and U2 bind the branch site, and the protein called U2AF, which is U2 accessory factor, bind the 3' splice site. U4, U6 bind to the U2 and U5, then the binding to the downstream exon, and then migrating to the intron exon boundary that make the intron RNA loop appear, as we see in the slide, and the complex then rearrange itself. In particular, U6 displace U1 from the U5 from the 5' splice site, and U1 and U4 are lost from the complex. The splicing process proceeds in two steps. So the first, the intron and exon are cut apart at the 5' splice site and free 5' end of the intron loop around, and is joined to the adenine at the branch site. Second, the free 3' end of the upstream exon displace the intron from the 3' splice site, and the two exons are joined together. The intron is released as a branched lariat structure, and later this lariat structure will be degraded. So that's the process of the splicing. And alternative splicing can create the protein diversity. So in this slide, showing the different exons can join with different combination, so create a protein diversity. And now we will watch the video of messenger RNA processing.
Before mRNA can be used by ribosomes as a template for building proteins, it must first be processed. Key steps are the addition of a methylated cap and a polyadenylated tail. Involved in processing are RNA polymerase, cleavage factors, and poly-A polymerase. Processing of mRNA begins with transcription. Soon after RNA polymerase begins transcription, a methylated cap is added to the 5' end. Transcription then continues to completion. Following completion, RNA polymerase releases the capped strand of pre-mRNA. Specific nucleotide sequences in the mRNA are bound by cleavage factors. The 3' end of the mRNA is next moved into the correct configuration for cleavage. Stabilizing factors are then added to the complex. Poly-A polymerase now binds to the mRNA and cleaves the 3' end. The complex begins to dissociate, and the cleaved 3' end quickly degrades. Poly-A polymerase now synthesizes the polyadenylated tail by adding adenine residues to the cleavage site. Additional proteins then bind to the tail, increasing the rate at which it grows. When the tail reaches its full length, the poly-A polymerase is signaled to stop adding residues, and the polyadenylation process is completed. The processed mRNA is now ready to undergo splicing in preparation for translation.
So that's the mRNA processing. just finished the discussion about the messenger RNA. And so about in the cell, 98% of transcriptional output of our genome is comprised of non-coding RNA. And the length of non-coding RNA can vary from 21 base pair to several thousands nucleotides. And these molecules can be divided into long non-coding RNA, such as LNC RNA, which are involved in epigenetic regulation of protein coding gene expression and modulation of gene transcription and protein degradation, and two small non-coding RNA, such as the microRNA, miRNA, small interfering RNA, siRNA, small nuclear RNA, snRNA, small nuclear RNA, snRNA, and both microRNA and the small interfering RNA are involved in gene expression at the post-transcription stage. The miRNA is endogenous to the cell and produced by transcription of the cell gene. It is about 22 nucleotide long and is cut from a larger RNA that contains a hairpin loop by the enzyme dicer. siRNA are from exogenous source, such as a viral infection or a synthetic double-stranded RNA. They can also be cut by the dicer to between 21 and 25 nucleotides. Although the microRNA and siRNA arrive from different sources, their mechanism of action are similar. So in the next slide, we will discuss the RNA interference by the siRNA and the microRNA. So the dicer is one of the RNA's three endonucleases. It takes the larger double-strand RNA and cuts them to their characteristic small size, leaving the two nucleotide long 3' overhang. Once the RNA is cleaved, it is passed to a protein complex called RNA-induced silencing complex, RISC. So both microRNA and siRNA can be cut by the dicer to the 21 to 23 base pair long siRNA or microRNA, and then the RISC will bind to the RISC will bind to the siRNA or microRNA. In an ATP-dependent process, the RISC unwind the double-strand RNA and select the antisense strand. Subsequently, a nuclease protein in the RISC complex known as the Argonaute will guide the complex to the target messenger RNA. In the case of siRNA, the matching of the antisense strand to the messenger RNA is perfect. Therefore, the Argonaute protein cleaves the messenger RNA, and the messenger RNA is silenced. This is process of RNA interference. In the case of microRNA, the microRNA are transcribed by the RNA polymerase II from what are called the MIR genes, and the first intermediate transcript a hairpin molecule called pre-MIR, similar to the messenger RNA. This transcript undergo capping and polyadenylation at 5' and 3' of the transcript, respectively. Unlike messenger RNA, the microRNA maturation begins in the nucleus and finish in the cytoplasm. And so basically the microRNA also processed by the dicer RNA, and then processed by the dicer and produce 21, 23 base pair of the microRNA, and the RISC bind. And after the binding RISC, the process, and then they keep the antisense strand, and then the microRNA basically they can bind to the messenger RNA imperfectly, and then they can block the translation. So that's also mechanism of RNA interference by the microRNA. And in addition to the replacing the messenger RNA translation and triggering messenger RNA and degradation, the siRNA and microRNA can also repress transcription of specific genes and the larger region of the genome through the associating with different complex the RNA induce the initiation of a transcription silencing complex, which is RATS reach. And the antisense RNA strand within a reach target the reach complex to a specific gene promoter or large region of chromatin, and reach then recruit the chromatin remodeling enzyme to the promoter, and this enzyme methylate histone and DNA, resulting in heterochromatin formation and subsequent transcriptional silencing. And here with the video.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs or double-stranded RNA is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to the dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonauts and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
So that will be the end of the video and also the end of the chapter.